This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1039, recorded on August 25th, 2023. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dixon Despomier.
Thank you, Vincent. Hello, everybody. I look out my window and I see clouds. I see little spots of water on my window, indicating that it might be raining out. The temperature's in the low 70s. It is very, I'm sorry, I take that back. It went up to 80. It's humid as heck, uncomfortable, and thank goodness I'm indoors.
Also joining us from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it's 75 Fahrenheit, 26 Celsius, after 2 nights of serious thunderstorms. Last night we only had less than an inch, but a lot of thunder and lightning. And the night before we had 3 inches of rain and just lots and lots of the lightning that doesn't have thunder. We were talking about, is that what you call heat lightning?
Yeah.
But it was raining, so it was just wild. 3 inches in pretty short time. So
Thank goodness.
yeah.
Also joining
and
us.
I'm looking up our humidity. Sorry, 77%, and the dew point is 67.
So joining us from Austin, Texas, Rich Condit.
Hi, everybody. The weather is a little different here. It's officially 104 degrees. It's kind of maybe a little hazy or something, like high veiled or thin overcast or something weird. It's not a clear blue sky, but it's stinking hot and will remain so for the foreseeable future. I think we'll start to get a little relief in September. And just to, since you guys did the same thing, it's humidity is 29%, so that helps a little bit.
Also joining us from College Station, Texas, Jolene Ramsey. Welcome to TWiV.
Howdy, everyone. It's great to be here.
Jolene?
Here it's just a little bit cooler than in Austin, 103 degrees, but I think we regularly are competing for hot temperatures.
Jolene, did you get any of that tropical storm? I forget what it was called.
Yeah, it was really windy here that day, and I couldn't figure out why until I realized there was a tropical storm.
Did you get any rain?
No rain.
Yeah.
No.
We got about 10 minutes of rain. I was in the theater watching Oppenheimer at the time, but
missed it.
we got about 10 minutes of rain, and that was good. And it dropped the temperature down to 80 degrees for 30 seconds. Back into the frying pan.
Rich, thank God it was not a thunderstorm. Otherwise, you would have thought maybe Oppenheimer is practicing his art.
Yeah.
So those of you watching the video, you can see behind Jolene, there are trees out the window.
Yeah.
We've been seeing birds land there. It's pretty cool.
Real trees.
Yeah, we've never had some wildlife outside anyone's window. So
Are those live oak by any chance?
Yeah, it's the live oak, some of the oldest ones on campus. It's a really nice view.
cool. Well, Jolene is the person who has done the timestamps on multiple podcasts for us for years. And she will tell you her history, but she works on bacteriophage. And so we thought we would have her come periodically to TWiV and tell us about her craft.
And she helped organize 2 TWiVs that we've been at. The one at Indiana University, there was Vincent and I were there, and then the one at College Station.
Yeah. Great events.
So we met at an ASM meeting. I don't remember where it was, but you came to a TWiV, I think, right?
I did. I came to, I think the first time we met actually was at ASV in 2016, I believe. And then I've seen quite a few live recordings at ASM.
This was your mom, right?
That's right. My mom made those pillows.
Behind me, too. Yeah, it's right there.
She's an avid sewer, and she did that as a favor to me. She prefers to make bags, but she paper pieced that with the help of someone who made the pattern. So
Sorry, I interrupted your other rep.
that's okay. I have met you guys in person several times. It's been quite a pleasure, and I've been listening for a long time, since 2011, when I needed to find something to listen to when I went to grad school and had to commute a little ways, and it's been great.
So I last saw you at ASM, right? Is that correct?
Yep, ASM in Houston.
And I came to your talk, and it was terrific. And so I said, you ought to come on and talk phage now and then, because we don't really do much phage here, because we don't know much about it, if anything.
I last saw Jolene maybe, 3 weeks ago?
Yeah, something like that. Mm-hmm.
Jolene and her husband came to Austin for a little getaway, and we had lunch together.
Cool. Very good.
Cool.
Nice.
All right, we're going to hear Jolene's story, but first, a couple of announcements. If you like what we do here on TWiV and all the other podcasts, I think we have 9 different podcasts now, and with more to come, we'd love to have your financial support. We don't do ads. We don't monetize our videos. We just depend on your support. It's always been my thought that ads are disruptive to learning, and I think we could do this on your generosity. So go to microbe.tv/contribute and help us out. Also, another announcement, there's a position available in Amy Rosenfeld's lab at the CBER division of FDA. This is for a research assistant to work on various picornaviruses like enterovirus D68, poliovirus, human rhinoviruses, genetic, biochemical, and cell-based studies for understanding pathogenesis, experiments in cell culture, also development of small animal models, all BSL-2 work. And if you want to know more, there will be a link in the show notes and an email where you can contact Amy to find out more. I also want to wish Alan Dove a happy birthday. He certainly will not listen to this episode, but today is his birthday, August 25th, and Kathy put it in the show notes, and then I looked at my calendar, and I had it there. I have everybody's birthday. I have to add yours now, Jolene, to my calendar. So happy birthday, Alan. Alan is in Mallorca. It's cool. Put a nice picture in the
yes, you did,
in the show notes. I also want to shout out to the grad students at the University of Vermont, which I visited this week, Tuesday, Wednesday, and Thursday. I was at their summer retreat, met a lot of them, gave a talk, and then we did a TWiV on Thursday, which was a lot of fun. And so thanks to them for inviting me and for talking with me. It was pretty cool. Jason Botten was one of my guests on TWiV, and I had been there 10 years before and did a TWiV with him and Marcus Talley. And Jason said to me at dinner, he said, you know, when you came last time, I showed you my virology course quiz, and you said to me, dude, what are you doing to them? This is too hard. You have to make it fun for them. You want them to learn. And so he said he changed them all. And I totally forgot saying it. I felt bad that I would tell someone to do with their course, but I do feel that you should encourage people to learn and have fun, not to bust them. It's too much. He was asking too many details, and I said, no, you don't want to do that. But he remembered it 10 years ago. So anyway, good visit. That podcast will come out over the next weeks when I am going to be absent a lot.
Where did they have their retreat, Vincent?
Right on campus.
Oh, okay.
Oh, there was an alumni center right there on campus. A very nice.
I thought I picture a lake, with canoes.
Well, there is Lake Champlain right next to it. Right?
Yeah, that's a pretty rough lake, though. That's
yeah, but and in fact, the hotel they put me up at was right on the lake, and then
nice.
It's all uphill to the.
Yeah.
They said, we'll send a car for you. I said, no, I'm going to walk. They said, it's a mile uphill. I said, it's fine. And they said, you'll sweat. I said, don't worry about it. And I did sweat. It was really, it was a tough walk, but it's good to walk, right? But walking downhill is hard too, especially at the end of the day when you're
That's
tired.
very true. Very
Wow.
true.
Anyway, thank you, everyone at the University of Vermont. Hopefully, it won't be 10 years getting back there. I met people from all over, and there were people from Michigan there, Kathy, from one
Oh, nice.
way or another, either faculty or postdocs or students who had passed through. Everybody seems to go through. Oh, I met a student from Michigan State, too. Okay, Jolene will do a little snippet for us today. Before you do that, Jolene, give us your history.
Yeah,
all the way
sure.
back, as far back as you can remember.
I'll go even further back than I can remember, actually. So I was born in California, Northern California, in a rural area, and I grew up there, moved around a lot in that area, and did not go to the same school for more than 2 years until I was in high school, and then that was still moving around, and then my mom moved my family to Colorado when I was 16, so I finished high school there in Colorado and went to Western State College of Colorado. It's now Western State University, Western Colorado University, I think actually it's called. And while I was there, I was convinced to become a biology major by my interest in the subject and by my professors who thought I was doing well in the classes, and by my then boyfriend, now husband. And while I was there, I also started doing research in a lab. I was actually working in the library because I thought I was going to be a librarian, but the librarian told me I should do research with her husband, who was one of the molecular biology professors. So I decided to try that out, and we were working on a bacteriophage called RB69, which is a close relative of T4. We were trying to solve the mystery of the R2 genes and what their proteins do, and we did not solve that mystery. It remains an unsolved biological mystery, but the R2 genes were really important because they were used as markers for mapping a lot of fundamental principles of how the codons are set up and other things like that. So I was playing around in the lab and enjoying it a lot. And then I did an REU for a summer. My advisor suggested, this was Dr. Peter Gauss, he suggested that I should apply for a summer research program. And I applied to Indiana University, among others. And then I went there to work in Eve Brune's lab for the summer of 2009. While I was there, I worked with a talented undergraduate mentor, Ellen Weinzapfel, and her postdoc mentor, Pam Brown, as well as Eve himself. And I was working on an interesting stalked bacterium called Astacoccalus biprosthecum. We were screening for genetic determinants of adhesion. So I spent the summer doing biofilm assays and PCR. And that summer, I realized that science was full of people because Western State College of Colorado was a school with 2,300 students. And when I was working with Dr. Gauss, I was the only student in the lab. And I enjoyed that very much. But at IU, there were many people in the lab all the time, and science was moving quickly and we were making discoveries. And I really enjoyed that. They encouraged me to apply for graduate school. I wasn't convinced right away that that was the best thing for me. So after I graduated from college, I decided to work in a lab. I moved to Denver and got a job in the lab of Ray Zhao. She's a crystallographer who had trained with Michael Rossman, and we were working on the proteins and RNAs that are involved in splicing in eukaryotic cells. I spent that year learning how to purify protein, purifying from liters and liters of yeast and bacteria. It was extremely useful skill development for me. And after not too many months of working in her lab, I realized that I really did want to have more independence in the science that I was going to be doing. So I decided to apply for graduate school that fall. And I ended up going to Indiana University, where I had done my REU, and did my PhD with Tuli Mukupidai, who works on alpha viruses. And I spent my time trying to figure out the roles of two different small membrane proteins that Synbis virus makes and how each of them are translated from the same gene, but they are translated by translational frame shifting. So they have the same N-terminus, but a different C-terminus. And it turns out that one of those forms of that protein has a different modification, and that results in it being packaged into the virus where the other one stays inside the cell, giving them different roles. I really enjoyed my work in eukaryotic virology, and that's where I started listening to TWiV, and I became more aware of a lot of the different viruses that were out there. But I still really loved phage and my original research that I had been doing, and I wanted to return to that. So that's what I did for my postdoc. I came here to Texas A&M to work with Rai Young, who is the world expert in how phage kill and get out of the bacterial cell at the end of their infection cycle. And I enjoyed my time in Rai's lab because I was doing teaching of undergraduate courses while I was doing my research. We were running a phage discovery, annotation, and publication course for seniors and juniors. I ran the lab portion and mentored many undergraduates through the annotation of their genomes and published those with them. And I was also teaching in a course with Jim Hu, where we taught undergraduates how to read papers and extract biological information from them and put that into a format that can be mined in a database and that is available through the gene ontology for scientists around the world to access. And I was also doing molecular work on phage lysis. And then in 2022, I was offered a position here at Texas A&M in the biology department. And so I moved over here and started my lab. And now I have 2 PhD students, a postdoc, a whole bunch of undergrads who do phage hunting, among other things. And so we study phage lysis, the mechanisms of how they get out of the cell, and just different ways that phage manipulate the bacterial host.
And when
What
you
a
were
nice story.
at Indiana, that's when you had Kathy and I come and
That's
we did
right.
a TWiV and a TWIM while we were there, right?
Mm-hmm.
There's a squirrel. Yeah, there's a squirrel behind.
There's a
Yeah,
bunch of them.
that's Rocky. He's getting his afternoon exercise.
Yep. All right. Thank you, Jolene. And you are doing our snippet today, so go for it.
That's right. Okay, so today I picked a paper that's actually about bacterial defense. So this is defense by the host against the phage. And the reason I picked this paper is because they did a lot of really cool work, and anti-phage defense is a major area of development in the field right now. So it's hard not to see lots of these kinds of papers. This paper is called Direct Activation of a Bacterial Innate Immune System by a Viral Capsid Protein. It was published in Nature in December of 2022. And the authors are Tong Zhang, Hedvig Taman, Kaio Kopieters-Wallant, Tatsiaku Kurata, Michelle Leroux, Sriram Srikant, Tetiana Bradziazenko, Albinas Sepauskas, Ariel Talavera, Chloe Martins, Gemma Atkinson, Vassal Hariliuk, Abel Garcia Pino, and Michael Lau. And in this paper, they are looking at the question of how do bacterial defense systems know that the cell is infected. There are some defense systems, such as restriction modification systems, that are on and active and patrolling all the time. And the reason they're able to have those on all the time is because there's a way for the cell to distinguish between its own DNA and incoming phage DNA, and that is the modification on the molecule. But in some cases, there are going to be proteins that are patrolling, but only activated in the presence of an infection, and it's unclear what aspect of an infection is triggering that defense system to come on. So that's the main question that they had in this paper. And they were looking at something called a toxin-antitoxin system. In this type of system, there are usually two proteins, but sometimes a protein and an RNA that are partners. And one of them is a toxin that is going to have an activity that can be toxic to the cell. And the other one is an antitoxin, which can bind to and prevent the activity of that toxin protein. So here they were looking at a type of toxin-antitoxin system where those two parts of the system are fused together in one molecule. And they thought that the conformation of the molecule was thought to be what was regulating its activity. So the first thing that they did was they found a system where they could take this two-piece toxin-antitoxin protein and split it apart and express each of those protein domains separately on a plasmid. And they did a few different assays, but what they can show is that if they express the toxin domain of the protein by itself, it will prevent the cells from growing. And if they express that with the toxin domain and the antitoxin domain together, then the cells are able to survive that overexpression.
What is the mechanism of the toxin? How does it prevent growth?
So that's coming up later in the paper, but what this particular toxin is it's an enzyme that modifies tRNAs and prevents them from being used in translation.
Wow, we did a paper on that recently, didn't we?
Yeah, this is echoes of last week. Where
cyclic nucleases. Right?
Yeah, where there's a what is effectively a toxin that is in an inactive form in the cell that gets activated because it somehow senses the infection and then targets the translation machinery via tRNA. I commented last time that was a Rube Goldberg way of doing things, but apparently not.
Yeah.
It seems more and more common.
Right. It is actually. And I think it's common because translation is a very important aspect of the cell that the phage is using immediately upon infection. And the mechanism here is that the phage come in and they deliver their genome. Then they begin to make proteins, and some of those proteins are going to get recognized. So what they showed here is that first they were able to demonstrate that this toxin-antitoxin, which is called capsid, that actually is encoded in the bacterial genome by a prophage, which is a phage that has been integrated into the host chromosome, is able to defend its cell against a variety of different phages, and different capsid proteins from different sources don't defend against the same phages. They, however, are therefore specific.
This is a wonderfully layered thing where you have a prophage. It's not the bacteria doing it.
Yeah.
It's the prophage. It's established a home there and is saying, all right, this is mine. Stay away.
Right. Exactly. It's providing a way to prevent superinfection, as we call it, superinfection exclusion, prevent other invaders from taking over that cell. So what they first looked at was this protection that they saw against phage. Was it going to be protecting the cell in such a way that it would reduce the amount of phage that got produced from a population? And they did see that there were fewer phages coming out in the presence of this capsid toxin-antitoxin. And they could see this with multiple different bacteriophages. They're using, in this case, T4 and something called SEC527. They also saw a similar thing with phage T7, and they found that there was a mutation in what they suspected to be the active site of the capsid protein that would no longer allow for protection against the phage. So they looked closer at this capsid protein, and they saw that they're in the two domains, the active domain that is going to be the toxin side, and then the other domain has a domain called a pseudo zinc finger domain. And this pseudo zinc finger domain is among a family of proteins that fold in such a way that they can bind to many different substrates. And exactly how long the fingers are and how far apart the fingers are is determined by what kind of cofactors and bonds are used to make the fingers themselves. So they thought that perhaps that was the domain that would be binding to sense something from the phage. And when they did something I love doing, which they chopped up the domains and mixed and matched them. So they took the Caprel protein from one source, split the domains, and mixed and matched them with Caprel proteins from another source. And then they saw which domain was conferring specificity to defend against a particular phage. And they saw that the pseudo-zinc finger domain in the C terminus of the protein was what was conferring specificity to defend against a particular phage, which makes sense because that's likely what would be binding to something from the phage. Then they solved the crystal structure of this protein in what looks like an open conformation. It looks kind of like a C with a tail coming up off of it. If you look at the structure of the protein, the zinc finger domain in what the conformation that they solved it is kind of out and away from the rest of the protein, and the active site where they had seen that mutation that was important in conferring activity is open and would be available for binding to something. And then what they did was, I thought, a neat technique that I've seen a lot recently, where they, even though they had solved the actual structure of the protein, they put it through this prediction algorithm called AlphaFold, and they looked for multiple different conformations of the protein, and they saw that there was a conformation that moved that pseudo-zinc finger domain down to block the active site, and that seems to be what they're calling the likely inactive domain, or inactive conformation. And then they were able to test that prediction later and show that indeed the conformation of the inactive predicted structure is consistent with what they see interacting with the phage.
So I have a question here. It
Yeah.
looks to me like, at least at a course level, the same zinc finger domain has two functions. One is to block the active site in the absence of the activator, okay? And the other is to bind the activator. But those, I'm thinking those are probably subdomains inside the zinc finger domain, because they got this little loop sticking up here that looks like it would be a candidate for binding the activator. I'm making this up, but what do you know?
Yeah. So further forward in the paper, they did actually find several locations, several specific residues that were important for binding to the phage molecule, which it turns out is the capsid protein, that are different in position from the ones that are binding to the active site region.
Okay, cool. That makes sense.
Yeah, I think the dramatic conformational shift allows it to have multiple surfaces that are going to be interacting with the different parts. And there are if you mutate one side, then it can still have its activity of blocking, even if it's not able to bind to the substrate. Okay, so the next thing they wanted to figure out was, how was this working? How does the toxin actually work? And how does it detect the phage in the incoming and infecting phage? The first question was, is it a matter of the protein, the toxin, antitoxin domains get separated, they get cleaved apart? And so they looked at total protein in the cell, and they see that it maintains one size at all times, both when it's in the presence and in the absence of a phage that it can block, suggesting it doesn't get cleaved in the cell. And then what they wanted to do was use our nice genetic tricks to look for an escaper or a suppressor mutation in the phage that was no longer able to be blocked by the toxin-antitoxin. But it turns out they had to put in a little bit more effort than we normally might have to do. So they had to passage several times. They took the population of phage that survived, which was very, very small compared to when Caprel wasn't present, and they infected a new population of cells expressing Caprel, and they continued to do that through 13 passages until they were able to grow the phage up to the same titer that they had had before they started the passaging. And then they sequenced these. They had 5 independently evolved lines. And when they sequenced them, 10 different isolates from those 5 lines, they found mutations just in 2 positions in the same gene. And therefore, that gene was encoding what they suspected to be the protein that would be binding to Caprel. And initially, that protein did not have an annotation. So when they looked at the predicted structure, it turns out it looks very similar to the HK97 capsid protein. HK97 is a very well-studied phage that has a nice, easily recognizable capsid fold. And this GP57 protein from their evolved phage has a fold just like HK97. So when they expressed just that protein from a plasmid in a cell with the Caprel protein, then they were able to prevent cell growth. And if they introduced the mutations that they had found from their evolved lines into the gene for GP57, they no longer had the cells dying from the toxic effects of Caprel. Then they started to look at what was Caprel doing once it was activated by this GP57 capsid protein. And they used a radiolabeling method to see that there was no longer much translation occurring in the cell. And therefore, they started pursuing this idea that it was a translation block. The translation block was effective in a plasmid system. They also saw a translation block in a phage infection. And then they even did it in an in vitro translation reaction where they put in a particular substrate to be translated. They put in purified protein and they saw that they were able to block the translation of that substrate. And then I think one of the nicest pieces of data that they put into the paper is just figure 3 panel K, where they took bulk tRNAs isolated from bacteria and they incubated those with purified Caprel, purified GP57 capsid protein, and a substrate ATP that was radiolabeled. So if they saw the transfer of the radiolabeled phosphate groups from the ATP to the tRNAs, then that would demonstrate modification by Caprel via the mechanism that we were just talking about earlier, where it's pyrophosphorylating the nucleotides in that structure. And that is what they saw. They saw that only when they had the wild type proteins mixed together did they get modified tRNAs. And they did not see that when they had the mutations in the capsid protein. So that was the nice piece of data about the mechanism for potential translation blockage. They only looked at bulk tRNAs, so it's not any more specific than that we know.
Do we know from other examples, can we anticipate whether there would be any particular species that would be phosphorylated, and within a given species, are there multiple phosphorylations or just one? Do you have any insight into that?
It's a great question. I do not know, actually.
Okay, fine.
Yeah.
And the phosphorylation of the tRNA is at a particular end. Is that relevant functionally, or just enough to mess up the tRNA?
That's also a good question that I do not know the answer to, but I imagine that is actually known.
Okay.
Got it. So this inhibition is enough to stop the infection, right? What's the effect on the host?
That's a good question. They didn't really look at that here in this paper. There is some some discussion in the field right now about whether or not these kinds of metabolic shifts that are induced by infection are really going to cause the host to be permanently damaged, or if it is a temporary induction of translation inhibition.
Yeah, whether it's altruistic or not.
Right, exactly. Is it really going to lead to cell death and therefore prevent the phage from coming out and protect neighbors? Or is it trying to only protect itself?
One of the things that interests me is that if, is the use of the major capsid protein, because that's going to be pretty late in infection.
Yes.
So you're cutting it close here, okay?
Cutting it close,
You've made capsid protein. By then, you might have enough stuff together so that you could actually put together some phage, but I guess not. Okay, or at least it's the yield is reduced because the biological experiment they have here is they do this low multiplicity infection where they show that under these circumstances the infection doesn't spread.
Yes, that's right. However, they also do the translation block. If you look in figure 3, panel I, that's a phage infection. And they do see a translation block, but it's not complete.
Right. Okay.
Because but it's not complete. And the other thing is, in some of their experiments where they're showing the dilution series of plaques on a plate, you see a major reduction, many logs reduction, protection, but it's not complete. There are still phage coming out. Yeah. Okay. So then briefly, I'll just summarize the last couple of figures. They went in to look at the nature of how the capsid protein is interacting with the Caprel toxin protein. And they did some biochemistry to look at they did pull downs, and they saw that the proteins seemed to be directly interacting with each other to form a complex. They also did several other assays that allowed them to determine which regions of the protein were more accessible. And they used that to infer where the two proteins were interacting. And that allowed them to find several residues that seemed to be involved in the interaction. So they found this position L280 and L307. And they changed those in the Caprel protein. And they showed that they were able to then change the ability to interact with the capsid protein. So they found positions both on the capsid that could change and prevent interaction. And they've now found positions in the toxin that would, the antitoxin domain of the Caprel protein that would prevent interaction. They no longer form a complex, and therefore there's no longer protection. And then they expanded this out into different phages, and they were able to look at the conservation of these regions in the capsid protein as well as in the Caprel protein. And they were able to show that for capsid proteins that were identical except for 5 residues, they were no longer able to protect. But if they changed the residues, then they did. And that overall led them to propose a model where the phage will bind to and deliver its genome into the cell. Inside that cell is already pre-positioned a Caprel protein that is in its inhibited conformation, and it's not active. Once the major capsid protein GP57 is translated, then it will bind directly to the antitoxin domain of Caprel, which will release that self-inhibition and allow it to phosphorylate the tRNA, which inhibits overall translation in the cell. That shuts down the ability of the phage to make more protein, and it can no longer increase the amount of assembled phage that are going to be produced out of that cell, and the cell itself will not be able to continue translation. However, what happens after that is not entirely clear from here, so I think that's an interesting future direction to be looking at.
It's kind of fun to look at this diagram that they have at the end, sort of the model, because you could think of the Caprel protein in the inactive state as just sort of a grenade or a little bomb. Mm-hmm. And then when the capsid protein binds to it, it's like pulling the pin out of the grenade, and now the bomb is going to go off because it's going to phosphorylate the the tRNA and then stop translation. So
Yeah,
I really like that model.
that's a nice way to look at it.
So there's a sentence in the discussion here that this paper for me is a major blast from the past. And there's a sentence in the discussion here that says, for PIF-A, which allows the F plasmid to exclude T7, escape mutants mapped to the major capsid protein, but this interaction has not been studied biochemically. And there's a reference to a paper. The first author is, and probably the major author is Ian Molyneux from University of Texas from 1989. At any rate, that F plasmid exclusion of T7, that was my PhD thesis. Okay, so
Wow.
I spent
Cool.
I spent years making baby steps towards understanding that. And interestingly, when I got that problem, it was based on previous research thought to be a translation inhibition phenomenon. Okay, and I did appropriate experiments to come to the conclusion at the time that was probably not it. It was something much more catastrophic. But I'm starting to think now that the catastrophe that I observed might actually in the end prove to be secondary to maybe a translation defect. Okay, who knows?
Yeah.
I don't think that problem is solved yet. And that's the other wonderful thing about this paper. What a rich environment this phage stuff is and how much there is to learn. Jolene, I'm envious. Okay. There was always a part of me that lingered in the phage world, part of my spirit. Okay. So good on you. It's great.
How is Jolene? How is it? Is it hard to do phage work? Is it hard to get it funded?
Is it hard to do phage work? No. No, it is not. We have a lot of great tools. They have small genomes, and the host is really based on the host, what makes it hard. So if you're working on a host that is an anaerobe or tricky to grow or something like that has very specific conditions, then that will make it harder. And if you're working in a host that doesn't have genetic tools, that makes it harder, but it's still possible. Can you get it funded? Well, I'm banking on yes. I have not yet successfully competed because I'm relatively new for external funding for this work. But I think it is because there is so much that we can learn, so many tools we have available, so many interesting questions, and there is a huge interest in the use of phages for different technologies. Whether people are interested in funding it for the study of phage itself, maybe not, but for the use of phage, either to treat antibiotic-resistant infections or in other applications, yes, definitely there is a place for that.
I'm always amazed. I see papers like this. Occasionally, I'll do them on TWiV, and I always say, there's so much to learn. It's amazing. Tools you have now are great.
I think there's some, as you were saying, some really incredible parallels between what we learn in bacteria about the mechanisms of defense against viruses with the things that we learn in eukaryotic virus systems.
Yeah.
So there's a lot of similarities, not at the sequence level, but in the principles of how the proteins interact with each other, how the hosts defend themselves, and how the viruses get around those defenses.
For sure. All right. Thank you, Jolene.
Yeah, really
Excellent.
nice explanation
Thank
of
you
this
very much.
paper.
Yeah,
Good job.
I agree.
All right. Our paper is a cell host micropaper called Widespread Human-Associated Redondo viruses infect the commensal protozoan, Entamoeba gingivalis. I've got a little bit for everybody here.
A little bit for everybody. That's
including
right.
Dixon.
Yay!
So commensal is something that commensal organisms, one of them is neutral and the other one takes something. Is that right?
Commensal means neither the host nor the organism gains much from the association.
Yeah.
The organism gains a house. The host, they've tried for years to prove a pathogenic role for Entamoeba gingivalis. It's found in normal gums and abnormal gums, and there really isn't a specific pathway for this organism to become pathogenic. It just sits there and eats bacteria. Basically, that's what it does.
Yeah, so I made a definition this week, and commensalism, one species benefits, the other is neither harmed nor helped.
Yeah, that's close enough. That's close enough.
All right. So this is from University of Pennsylvania. Emma Keeler, Cardo Merenstein, Shantan Reddy, Louis Taylor, Anna Kobian, Guemes, Orvi Zankaria, Ron Coleman, and Fred Bushman, who was on a recent TWiV. Fred Bushman. Redondo virus. This has been in my TWiVable file for a long time, and I thought Jolene would enjoy it as well, because it's got some the word phages scattered throughout. So
it's got a lot of parallels to phages.
It does have a lot of parallels, yes. As you will see, redondo viruses were first found as dark matter, which was a term that Eric Delwart told Rich and I years ago when we went out there to do a TWiV. Remember, it was the first time we heard that.
Absolutely.
You sequence DNA, you don't know what it is. You call it dark matter. And this was found in virome samples of the human respiratory tracts. You take out material, you sequence it, and you get some where you say, yeah, this virus is here, and some you have no idea, so it's dark matter. They found in a few samples from lung transplant donors and recipients a few sequence reads that were aligning with a porcine virus, porcine stool-associated circular virus 5, identified in pig stool, obviously. And these turned out to be circular DNA genomes, about 3 kb, which encode proteins very similar to those of the capsid and replication-associated proteins, the cap and rep proteins of known single-stranded DNA viruses that are called CRESS viruses, circular rep encoding single-stranded DNA viruses. And these dark matter was eventually put into a new family of viruses called redondoviruses, from redondo, the Spanish for circle.
Is this rep?
Actually, wait a second. It's not Spanish for circle. It's Spanish for round.
Is it? Okay. Thank you. I wouldn't know that, but they say it here in the paper.
I know. I marked it.
So why did they say circle?
I'm not sure, because even in other places where they talk about redondo viruses, they correctly attribute the meaning. So, yeah.
Let's see. Are there any people who
Circles
should know better
are
on this paper?
They are.
Yeah, they are round. I guess it's
Rondo
close enough.
is a musical term.
What's a Spanish word for circle?
Círculo.
Oh, we already have circoviruses, so that's
pretty intuitive.
No, but we have circoviruses, so probably they didn't want to. Oh, okay, thank you. Anyway,
so
do
you know if the rep gene in these things if the protein is related to the rep protein in parvoviruses, or in anything other than by name?
So, no, I think in this paper they show some biochemical properties that are similar to
biochemical properties. Yeah,
yeah, that's what I don't know about homology.
Okay.
Yeah. So anyway, there's 2 species in this, and I'm not going to tell you what they mean because they're probably wrong, right? Jolene?
No, they're right, actually.
They're good. Viento virus and Brisa virus.
Mm-hmm.
Viento, wind, and Brisa, breeze, right? That's good?
Yep, that's right.
Okay. And here we go. The purified rep protein has the expected enzymatic activities for a rep protein rich.
Okay.
All right. So these eventually people started looking at these redondo virus genomes more, and they found that they're present mostly in human samples from the respiratory tract, the mouth and the respiratory tract. They're found in healthy people from all over the world. Prevalence 2 to 80%, depending on the people in the samples. And when people have certain illnesses, they're found at elevated levels, like lung transplant donors and recipients, febrile patients, people with periodontitis, rheumatoid arthritis, inflammatory bowel disease, et cetera, even COVID and AIDS. So that's redondo viruses, and a bit of a mystery. So here they want to know, all right, they're in people, because we get samples from people, we sequence them, and we find these genomes. So where are they reproducing?
Yeah. Also, they're elevated under circumstances, under disease circumstances.
Yeah.
So the question is that causal or not? Okay.
Yeah.
Are they actually pathogenic? Okay.
Right.
Yeah. We'll come back to that.
So they don't appear to replicate in prokaryotic cells. So this is where I said this would be a good paper for Jolene, right? Because the genomes don't have what's called the Shine-Dalgarno sequence, which is needed for initiating translation, which it would have to require if it reproduced in a bacteria. Right, Jolene?
Correct.
Okay, I got to make sure now we get...
It's funny, though, because they say they don't have this Shine-Dalgarno translation initiation that is characteristic of bacteriophages. And I was like, duh, what about bacteria?
Bacteria too, yes.
I think they're trying to say that it's not a bacteriophage because it doesn't have commonalities, but you're right.
Right.
And furthermore, these Redondo virus sequences have been found in CRISPR spacer arrays, which are produced from viruses that would infect bacteria with CRISPR systems, and they would store bits of them in the genome, and you don't find that there. They're also, their genome sequences are distinct from single-stranded DNA bacteriophages, of which there are some, of course. So they say, well, maybe they infect human cells, but they're going to provide evidence in this paper that they actually infect amoeba, Entamoeba gingivalis, that are in us. And there I figured Dixon would like this too.
Well, I like everything else too, but I'm particularly partial to eukaryotes. Let's put it that way.
No, I try to please everyone. Well, actually, I don't, but this happened to be an easy paper for that. I just look for interesting stuff.
Yeah, right. So to point out the parallel, so the bacteria in us have a virome that is bacteriophages, and now we're going to get hints that the amoeba in us have viruses that infect them, and they could be like this.
So Jolene, every part of us has some kind of microbiome, right? Mm-hmm. Like every organ system, even our skin, right?
Correct.
And with that, we have a virome consisting of bacteriophages, correct?
We have a virome consisting of eukaryotic viruses and bacteriophages.
Correct.
And we also have archaea in our microbiome. Therefore, we will also have the viruses that infect archaea, which you may choose to call archaeal viruses or phages, depending on who you are.
Oh, gosh. The guy in Portland would not like them called phages. What's his name?
Ken Stedman.
But they're not bacteria, right?
The archaea?
Yeah.
Correct.
So why would you call them bacteriophages?
You would just call them phages. Exactly. Archaeophages.
So that's an interesting question, right?
Because
we call bacterial viruses too, right? We don't have to call them.
Well, you call them bacterial viruses, and I call them bacterial viruses, probably because I was trained in a eukaryotic virus lab. However, the people who have studied bacteriophages for a lot longer don't necessarily call them bacterial viruses.
But there are viruses, though. No doubt about that, right?
Correct.
Yeah, I like archaeal viruses. Because, well, it depends who the first person who discovered them called it, right? I don't know who that was.
Well, they thought that archaea were bacteria, so...
Yeah.
Right, exactly.
All right, so what kind of evidence do you get to show that these redondoviridae, members of the redondoviridae, reproduce in Entamoeba? All right, so the first thing they do is get a lot of genomes of redondoviridae. They look at the rep protein. There is a phylum that contains these CRES viruses. It's called CRES DNA viricota. I don't know if you pronounce the DNA separately or Cresdena viricota. I don't know. CRES DNA viricota.
It's like a dessert. It almost sounds like a dessert.
There's someone on Instagram with DNA viricota as a name. Every time I see it, I have
a cup of DNA viricota just before bedtime. I wake up, I feel great.
So these sequences are annotated with probable host organisms, and so in these trees, they construct phylogenetic trees. a close neighbor to the Redondoviridae family, is the Nariaviridae, whose members have recently been proposed to reproduce in Entamoeba. Okay, so that could be some evidence that the Redondoviridae members reproduce there as well. These sequences are, as I kind of mentioned, they're associated with humans for sure. They're represented highly in sequencing data from samples from the gingival crevice of periodontitis patients. And the amoeba E. gingivalis is the second most abundant in terms of ribosomal RNA in such samples after human. A lot of sequences there. So
I'm going to presume it's also an anaerobe.
The
Because
Entamoeba?
all the Entamoeba that I know of are anaerobic. So they're susceptible to a drug which takes advantage of that.
Is the crevice not anaerobic, but
yes.
They don't have any
Yes.
They don't have any mitochondria.
It is.
It's anaerobic. Yeah.
I had to look it up for anybody else who doesn't know. I assumed, but the gingival crevice, also known as the gingival sulcus
pocket
or pocket, I guess, is the part right where your tooth is meeting the gum, and the gum is poking up, and there's kind of like a V-shaped spot where these microbes can find a nice little niche to exist, especially if you don't brush your teeth.
It's all about niche. I hope no one doesn't brush their teeth, do they?
Are you kidding?
It just maybe depends on the frequency.
At least twice a day. But Michael Schmidt was talking about that recently because, as you know, I'm sure you know this, Jolene, every time you brush your teeth, you get a transient bacteremia.
I've heard you guys say that many times.
And so Michael said it last week again. He said, so if you brush twice a day, which most people should, you have 12 hours of bacteremia every day. Isn't that something? And we did this on the live stream. A lot of people didn't know that. Another way of making a connection is by the percent GC content of the DNA. Right. And that number for DNA viruses often mimics that of their host. So they look this up. And the median GC of the two redondoviridae species, Viento and Brisa viruses, are 34.6 and 33.9%. And Entamoeba sequences are low GC as well, from 24 to 30. And our genome, human genome, is about 40.9%. So that's also consistent with E. gingivalis reproducing, redondoviruses reproducing in E. gingivalis. Okay. Another thing you can do is to see whether the sequences of the redondoviruses and the E. gingivalis, they co-occur in different kinds of samples, right? You find one, do you find the other? So they looked at 58 metagenomic data sets, 7,869 samples from different parts of the human body and disease states, and they found 4 data sets positive for redondovirus DNA, and these are all these include samples from the oral cavity of healthy individuals and people with peri-implantitis, mucositis, and periodontitis. A lot of words I've never heard before, frankly. And I don't want to say anything because every time we get in trouble. I got nasty emails last week about our discussion of blood clotting. Oh, my gosh.
Oh, right. Exactly.
However, we got compliments from one of the authors. So
no pun intended.
We did. So it all balances. Yeah. Right. And these metagenomic data sets from submucosal and subgingival plaque of healthy controls in patients with peri-implantitis or mucositis, the virus, redondoviruses, were highly correlated with E. gingivalis. So that is also consistent with the viruses reproducing in this E. gingivalis. Then they looked at co-occurrence of the virus and E. gingivalis in disease states that have been previously shown to be have elevated redondovirus prevalence. So previously elevated prevalence. Now they say, do they have E. gingivalis DNA in them? They do this by a qPCR. They look at oropharyngeal, nasopharyngeal, and endotracheal aspirate samples from 38 ICU patients. And of the 38 patients, 3 were positive for Redondo viruses, and all of them were also positive for E. gingivalis. Then they have another cohort of 88 patients hospitalized with COVID, where they have previously shown elevated Redondo virus presence. And again, positive association between in these COVID samples between Redondo viruses and E. gingivalis.
Right. 9 out of 10 of them have the E. gingivalis.
And then they looked in human saliva samples from 50 healthy volunteers in Philadelphia, probably all from UPenn, I bet. Walk down the hall. Can you give me some saliva, please? I don't necessarily know that, but of the 16 samples that were positive for Redondo virus DNA, 15 were positive for E. gingivalis DNA. Again, so that's all pretty good. The p-value is really good, Dixon.
Mm-hmm. I hear that.
Okay, and then next thing they did is to look in. Gosh, I would have just infected the things on day one, man. Okay, in metatranscriptome data from periodontitis patients. So they had 18 publicly available RNA-seq data sets, 1,879 samples from diverse body sites. So they looked to see, okay, are there Redondo virus sequences? And then in those, you find E. gingivalis. And 2 of these data sets, 2 of these 18 were positive. They both had samples from the gingival crevice of periodontitis patients. 2 of them were positive for both Redondo virus RNA, and they were positive for the viral RNA. So they looked at them further. And in the first study, they looked at the metatranscriptome of matched healthy and disease gingiva from 3 people with aggressive periodontitis. I get very squeamish about all these mouth and tooth diseases, I have to say. All 6 samples from both diseased and healthy sites were positive for E. gingivalis. Only the 3 samples from diseased sites were also positive for Redondo virus DNA. In another study, they had metatranscriptomes of pooled gingival tissues from both periodontitis patients and healthy controls. Both the healthy and diseased pools were positive for E. gingivalis, and again, the Redondo only found in the diseased pool. So that's all very good associations. And finally, what about doing an infection? Well, here we get, this is as close as we get. You can't grow E. gingivalis in pure culture.
That is right.
Did you know that, Dixon?
I did.
I have people.
It's very difficult to grow a lot of amoeba in azenic cultures.
Yeah,
because they have associations with their microbiome, and they need them in order to survive.
Yeah, so they say you need feeder bacteria in this case. The same with Entamoeba histolytica. Do you know, Dixon?
Yes, it is. It's very difficult to grow in alone.
You need live bacteria? What if you made an extract? Would that work?
You don't need live bacteria. You need, well, and Entamoeba histolytica doesn't feed on bacteria, by the way. It feeds on red blood cells.
So you put red blood cells in and you can grow it?
Yeah.
Too bad. Well, E. gingivalis needs feeder bacteria.
They do. Just like Cenobitis elegans.
So, Jolene, feeder bacteria, are they living?
Yes. Usually.
They are?
Usually they are. You grow them and they grow alongside whatever you're feeding them to.
Because there are some eukaryotic feeder situations where you irradiate the feeder cells and they don't grow anymore.
Let me double check the methods.
But anyway, so they got a culture of E. gingivalis which has bacteria, right? So they say they have Entamoeba trophozoites in it, and they show that there's E. gingivalis RNA and DNA, so this is the right thing. Gosh, someone said to me in Vermont, we have to sequence everything. Nobody trusts anyone anymore.
All the cloning that we did, before you would sequence things. You would do restriction digest, convince yourself that you had what you expected and go on. And now everybody sequences everything.
They were complaining this. We spend all our time sequencing things, It's amazing.
So in the methods, it says that they generated a medium that a sterile TYGM9 medium, which includes a rice starch solution, and then they bacterized it by adding supernatant that contained bacteria from the culture to another culture tube.
Okay.
And they don't specify which bacteria.
They do not. They do
Interesting.
specify that they're carrying out instructions from the ATCC because they got this from the American Type Culture Collection.
Right.
All right.
So I
So
would presume they were derived from the oral cavity rather than the anal area, which is where E. coli would be obviously found, and used as a dead bacterial growth medium for lots of other things, like Entamoeba, for instance, but other things too.
anyway, this culture has redondoviruses in it. So
Right.
they didn't add redondovirus.
Right,
It's just
right.
there. What they got from
It was-
ATCC was already infected, I
Are you trying to tell me it was redundant?
guess.
Redundant?
Redundant.
They say it was robustly positive, which is just we don't really like that. But there are a lot of copies of DNA, 2 times 10 to the 8th DNA copies per milliliter of culture fluid. And there are no human cells in this culture. They look by PCR for a human enzyme. They don't see it. So they say the virus must be replicating in one of the unicellular organisms present in the culture.
Right.
So they actually got the viral genome sequence out of this culture. They gave it a name. It's 3.162 kbp in length. It encodes cap, rep, and ORF3 proteins. It is a member of the Vientovirus species. And its nearest relative was isolated from a human respiratory specimen. Okay, so the next experiment they do is very interesting technology. I'm not sure I would have done it, but it's called high C. I think C stands for chromatin, right? Because you're looking at chromatin structure with it. I guess, Kathy, you put in the description. Do you want to describe high C?
You can describe it, but basically because they can't grow the amoeba, they're trying to figure out other ways, orthogonal ways, to show that the virus is associating with this.
Yeah.
And so, briefly, they're using a method that's going to allow them to cross-link the viral DNA to anything that it's nearby.
It's a cool method, yeah.
And so what they find out is that it's nearby to the Entamoeba.
Yeah, so, these cultures already have redondovirus DNA in them. They say, so can we cross-link the redondovirus DNA to the amoebal DNA by this technique that's typically used to study chromatin structure? And they do find that it's associating. So you have to do statistics because they get some cross-linking with Parabacteroides, a member of, this is a genus, right? So something in there. And does that mean, Jolene, that these are in the culture? I guess it does, right?
Yeah, they do metagenomics on their xenic culture in Figure 4, and they're showing that there's quite a range of organisms present, including E. coli, or at least Escherichia.
Yeah, Bacteroides.
Yeah, this is because they bacterized it, or what was that the word?
Yes, yes.
They bacterized their culture. Yeah. Anyway,
sounds like a Ghostbuster term.
Yeah.
The question still remains, though, as to whether the virus is in the bacteria or in the amoeba.
No, it seems to be in the amoeba because the cross-linking shows that you can cross-link it more frequently to amoebal DNA, right?
It would be very easy to. Well, where is it? You should be able to, under electron microscopy, then demonstrate.
Well, yes, I thought, why didn't they look?
Why not do that?
Why didn't they do in situ hybridization?
Exactly.
They addressed that. They've had the end of the discussion, they said they've had difficulties working with xenic E. gingivalis culture, precluded further investigations such as fluorescence in situ hybridization to co-localize them. I thought maybe something like flow cytometry would be a nice
that
way
would do
to
it.
separate them.
You should be able to separate the bacteria from the amoeba, right?
Yeah. Assuming
You
you can flow amoeba if they're not too big. I don't think so, right?
No,
They
you have
should
to
be
think
okay.
about what the amoeba eats. The amoeba is eating not human cells, it's eating bacteria.
Yeah,
So it's got bacteria in its food vacuoles.
yeah.
Mhm.
So that's within the cytoplasm.
I see.
So you're not going to get rid of that,
It's going to be a
even
problem.
if you
Yeah,
started with a single amoeba.
right.
They still would be able to see that the virus. You could see where in the amoeba it was.
Yes,
Yeah,
I
where
agree.
in the amoeba, right? Is it in the
You could.
vacuole where all of the other bacteria that
That's
have been
right.
eaten are, or is it
That's what I'm saying.
where the
That's
Redondo virus usually is?
Going back to the high C method,
Mm.
where they talk about finding the Redondo virus DNA sequence, they to address sort of Dixon's question, they find it associating 101 times with Redondo virus DNA, and then they find it 6 times associating with Entamoeba, and only 2 times with this one bacteria.
Yeah.
And then there's 5 examples, 1 per taxon, of some other bacteria.
Yeah.
So the predominant thing that they find the Redondo virus DNA associating with is this Entamoeba, other than more Redondo virus DNA, which
But Kathy, what I'm trying to say is that the bacteria are inside the amoeba and you can't dissociate them.
I don't think the bacteria are inside the amoeba
Why don't you think so?
in this case.
They are because they ate them.
Are they?
Yeah.
That is their food. So if you're going to starve out gingivalis, they will die. If you used antibiotics, for instance.
Well, then that being the case, wouldn't you expect if they were associating with bacterial DNA that you would find many more cases of the bacterial DNA associating with the Redondo virus DNA?
If the native host was a bacterium, then you would expect a higher proportion of the linked DNAs to be with that bacterium. Yes.
Right.
Yeah.
Right.
So the easy solution is to do an immuno-EM.
Yeah.
If
Maybe
you can,
maybe not so easy, but it is one approach. Yeah.
or a hybridization.
So I'm wondering if there are any gingivalis cells that are uninfected in these cultures, and if you could separate them out and grow them. Do you think they're all infected with
that's their food.
No, Redondo virus infected, not bacteria. That's not infected
Well,
anyway.
if the Redondo virus is inside the bacteria, you're screwed.
Well, I don't think they are actually, but there must be uninfected cells, and if you could stain a viral rep or cap protein, I know they have problems doing this, but come on, fix it, sort it out, and then they could stain. They could stain for uninfected cells and gate those out, and then maybe grow those, and then they could add virus and watch it reproduce, which I would like to see. I mean, because these cultures are hopelessly infected with Redondo viruses already, so adding more probably wouldn't give you a bigger signal.
And presumably, they should be able to control which bacteria they're adding to their media, so they could vary that. And
So you could do so if you single cell sorted and got Redondo uninfected amoeba, then you could add a single bacterium species.
Yeah.
So you keep it
what
simple.
If you didn't?
Right?
What if they were all infected?
I'd be really
then
surprised.
You have to try
actually.
something else.
I'd be surprised, right, if they're all infected. But it sounds like there's a lot of virus in there. So yeah, it's a high multiplicity. So.
There's a lot of statistical evidence, but there's no definitive proof.
It's all consistent. All these.
I'm trying to think how you could create a culture.
Yeah.
Maybe you need some Redondo virus antiserum or something like that you could stick in a culture of gingivalis and serially propagate that. Something like that.
So the Redondo viruses replicate in the nucleus, right?
Yeah. Yep.
So could you isolate nuclei from Entamoeba?
Yeah.
And separate those from the vacuoles with.
That's an easy deal. Use a sucrose gradient, and you can easily get them out.
Well, I think they would have done it. When was this paper published? It's
January this year.
this year, 2023. Okay, not
They
so
may
long
not
ago.
have, using sucrose gradient ultracentrifugation to isolate subcellular particles. That's the laboratory that I spent my time in at Rockefeller, and I watched them isolate peroxisomes and lysosomes and mitochondria and nuclei, of course, and other things too. And that's so that if there's a isopycnic point that's different between the nucleus and the virus, and of course there would be, and everything else inside the amoeba, it doesn't have too many intracellular particles actually, because it has no mitochondria.
Mm-hmm.
So and the nucleus is huge. The only thing you're worried about is contamination.
Wait a minute. Did you just say the amoeba has no mitochondria?
It's an anaerobe. Excuse me. It's an anaerobe.
What?
It's why it lives in the pocket of the gingiva,
Yeah. Okay.
where the oxygen
yeah.
content is 0. Okay.
I don't
Are
want
there
to.
any further questions?
Right. I don't want to.
They have mitosomes, Dixon. They have mitosomes,
They
which
have mitosomes. Yes. Okay.
they are
okay, but
mitochondria lacking some canonical functions.
well, they like aerobic respiration.
Yeah, well, that's fine, but there's still something that looks like a.
They live in an anaerobic cell.
Yeah, I got that. But all right, what about disease? What is is
Kathy had something to add on that, maybe.
Yeah, Kathy said we would get to it. You want to talk about it, Kathy?
Sorry, I was looking up something else.
Okay, so there's an association of E. gingivalis with disease. So it's a common inhabitant. It's in most of us, right? In the oral cavity, and they find it now and then in the lungs, and they wonder if it translocates. Maybe in disease states, it gets down there, but nobody's really looked at that. It could be, just a non-pathogenic colonizer, or it could contribute to pathogenesis or inflammation.
Vincent, when I was a graduate student at Columbia, our laboratory was right next to the dental microbiology laboratory.
Yeah.
And we talked, there was a lot of crosstalk between us, and they looked a lot at this, trying to pinpoint a role for this as a pathogen in periodontal disease, and they couldn't do it.
The amoeba.
Yeah.
Right.
Exactly right.
And so that is kind of what I was just now looking up to, because you're right, Vincent. In the discussion, they talk about maybe these Redondo viruses are just markers, even though they're associated with the disease states.
Right.
But there's some data that just now, as we were going through it, made me think that it argues against that. And that's when you talked about the fact that they had the samples from diseased and healthy states, and only the samples from the diseased states were also positive for Redondo virus.
Exactly.
Okay.
Yeah.
And that was 3 out of 3 and versus 0 out of 3. So
right.
the sample sizes are small. So maybe that's part of it. But, when I was first reading this, I thought, okay, so how is the presence of this virus going to make these amoeba more virulent? And so then I was thinking about the phage situation where we know that's the case. And is it Shiga toxin? Yep. And there's another
cholera
one too,
toxin.
I think. That's right. Okay. So those are cases where having the virus makes it more pathogenic. And so that would be a parallel here if that's the case. But I think there's just not enough data, maybe. I'd be interested to hear what you guys think.
Are the Circo viruses in general, do they have latent infections?
I wouldn't call them latent. They're reproducing, but not causing any disease in humans anyway.
In an individual cell, is the cell...
That's a good question.
harmed?
Is it lysing the amoeba?
I don't know. I mean, you can imagine that it might not. It might not lyse the cell, but still get out, or it could lyse in, a few cells here and there. It doesn't cause any pathology as a consequence.
Mm-hmm.
So a lot of these viruses, they're double-stranded DNA viruses, or rather
so they need to
polyomas.
Do
they're all reproducing in people, and they don't always cause any disease. So not clear what's going on.
Yeah, but it's different for viruses that infect single-celled organisms, right?
Oh yeah, of course. Yeah,
a lot. There's a lot of double-stranded RNA viruses that also infect flagellates, for instance, like Trichomonas vaginalis and things like that. And they do alter pathogenicity.
Yeah, they talk about that as a part of the these eukaryotic viruses of eukaryotic commensals, right?
Right.
And
Well,
they talk about Leishmania, Giardia, Trichomonas, Cryptosporidium, Plasmodium.
those are
and
not
then-
commensals.
Well, they called them. They put them in the same phrase, but
No,
I know they're not
no,
commensals.
they're
Yes,
wrong. They're dead wrong on that one.
They say
You
parasites.
know,
Parasites.
they do call them
that's
parasites.
fair. That's
yeah.
fair. That's fair enough.
But you can find some of those viruses of those organisms in human samples, right? But I think it's a good question. I would have to look it up. I don't know I don't know that anyone's done it. Infect E. gingivalis with these redondo viruses. They can't. They're trying to do it here. So I don't know what kind of CPE you would expect. It's a good question. I mean, in these cultures, Jolene, they're making a ton of DNA. They would see CPE, right? And they don't mention it at all.
Maybe.
I just don't know.
I mean, it
Or
depends
like
on
calling-
if all the cells are synchronously infected.
Yeah.
Ah.
Yeah, but so it could either be yes or no, right? It could go either way. And
You're talking about CPE in an amoeba?
yeah, the thing.
I mean, how can you tell?
Well, it breaks. It breaks up. No,
Oh, okay.
You'd have to, or
Yeah,
you get
lysis.
it shrinks.
infusion bodies.
we call it lysis or some other CPE. It could shrink.
Yeah.
It could
It's
make blebs.
biochemistry
I don't know.
change.
I don't know what they do, but they could at the one end they could lyse. At the other end,
Right.
they might not. I just don't know. A
So
lot of
they
questions
need to-
here.
They need to develop a
So-
cell-free azenic culture
So
medium.
let me get your feeling at the end here. What do you think? Do you think redondo viruses reproduce in E. gingivalis?
Yeah.
Rich? Yes.
Yeah.
Jolene?
I think there's compelling circumstantial evidence.
Compelling.
What do you think, Jolene?
I think probably, but there is some missing key things.
Mm-hmm. Kathy?
Yep.
I'm like Rich. I think it's pretty compelling circumstantial evidence. I mean, I would argue, what else could it be if it's not that?
Yeah. Well, it's probably, and as they say in the discussion, it's probably not the bacteria because there's no Shine del Garno. And then
Yeah.
you find a virus that can reproduce in bacteria without a shine. Who knows? Dixon, what do you think?
I think they need to do more research. That's what I think.
Well, they do, but do you?
No, they need to do more definitive research.
Yeah, I think
it's exclusive. Yes or no. Yes or no. No statistics. Yes or no.
I think
the cell sorting idea is a good idea.
I think
It's
it
a very
leans
good idea.
towards yes. I would like to see some kind of infection experiment.
Yeah.
Right. Where
And-
you either look at it
and-
by CPE or you show staining that there are viral proteins in the amoeba and not in the bacteria.
And EM. I mean, you could use a cDNA from
Oh, how about this?
circular
How about this, Jolene? You
hybridization.
take some of these bacteria that are used for the feeder.
Mhm.
You take those on their own and add redondo virus and see if it reproduces
right.
in them.
Mm-hmm.
Exactly.
They didn't do that. It's a negative result, but
Right.
No,
we
but
don't
you know
even
what?
we don't even have any redondo virus.
Come on, this could easily, Vincent. Remember the situation with Wolbachia, and Wolbachia lives inside of a vacuole inside of a eukaryotic cell,
right.
and then now you got a special environment there which you can't replicate in vitro rather.
So
Or you could, but they haven't done it
the
yet.
bacterial infection experiment's not good because there's no positive control.
Yeah.
That's right.
And you don't know which bacteria is the right one.
All of the above.
And so you'd have to infect every bacteria that's in this xenic culture, right?
I think
Not a great experiment.
the best thing would be to have a culture as a xenic, have
Yeah,
the xenic culture that did not already have redondo virus in it.
Yeah.
good
That
idea.
would be really helpful.
That's right. And then as the amoeba reproduced, maybe there would be more circular DNA in the supernatant, and that's evidence that it's coming from the amoeba. Because you've wiped the surface clean, and it's got to be coming from someplace.
So
right?
so not every human has redondo virus. So maybe you get some E. gingivitis from gingivalis from one of those Redondo negative humans and try and grow it out, right?
Mm-hmm.
Don't go to ATCC. They just have one line that's got virus in it. It's not good enough, right? All right. Anyway,
So,
go ahead.
Did they comment? They did, didn't they, on how common sort of amoeba are, in particular commensal amoeba in humans?
Yeah, they did.
This is like the only one or one of the
No,
few?
no,
Is that
no,
right?
no. You want a list? I can give you a list.
I'm not interested in hit and run. I'm interested in guys that are living with me all the time.
I am not everybody has commensal amoeba living inside of them, but many. I worked in a diagnostic lab when I first started my career at Columbia, and about 10% of all the patients that came in suffering from diarrhea had non-diarrheal causing agents in their stool that we could not identify by microscopy, so they sent them over to the microbiological laboratory. They still had amoeba that was proven not to cause disease. Iodoamoeba buchleri, Endolimax nana. There's a list of these, Entamoeba pollecki. All these organisms that you find that you can identify, none of those had been identified with disease states. None of them. But what they had been identified with is fecal contamination. Now, gingivalis is different because it lives in the oral cavity. But the other commensal amoeba are all transmitted by fecal contamination.
By the way, Jolene, there were 3 papers here that I thought were interesting. So they're 48, 49, and 50. Bacteriophage trigger antiviral immunity and prevent clearance of bacterial infection. Expansion of bacteriophages is linked to aggravated intestinal inflammation and colitis and bacteriophages in the immune system. So, those are cool.
Right. We can pick something in that vein for next time.
Yeah, it would
those
be
are
good.
Those are fundable catchwords.
No, the one where it's in aggravated intestinal inflammation and colitis, I'd like to look into that.
Well, it turns out that not only do bacteriophages interact directly with human cells,
Right.
the presence of certain bacteriophage correlate with the abundance of different bacteria that can be pathogenic and cause these kinds of things.
Right.
Do you know if you can do an in situ hybridization on an ultra thin section?
You mean
with
like
a fluorescence? Yeah.
No, not fluorescence.
You mean for an electron
using
microscope?
Yeah, that's right. Using like ferritin or something like that.
Well, you can do even a gold staining.
Yeah, that on a section.
Yeah.
That's what they should do. They should make a cDNA for the Redondo virus and label it with gold, and then in situ hybridize a section of Entamoeba gingivalis and find it.
Well, I think you could do it more simply with fluorescence, right? In light microscopy.
You could say it's in the cytoplasm, but remember, there are bacteria in the food vacuoles.
Right.
You'd have to say they're not in those.
Okay.
They're in the cytoplasm of the amoeba. That's
fine.
right.
All right.
That's right. Exciting paper.
It's cool. It's good discussion.
Sounds like a thesis to me.
All right. All right, let's do a round of email.
Sure.
Dixon, take the first one, please.
I thought you'd say that. Let me scroll down here. Email. Marcus writes, Dear TWiVers, during this episode, you referred a lot to boatloads several times, and at one point even asked, how much is this? So here goes my theory. Boatload is a euphemism for buttload. Buttload. People avoid to say buttload because they think it refers to a human posterior, but it does not. A butt is a standard size of barrel equal to 2 hogsheads. Oh, well, that clears up everything. The hogsheads varies in size, but today usually is taken to be 63 U.S. gallons. So a boatload is 106 gallons, or much preferably to me, about 4 hectoliters. Have you ever heard of that? Hectoliters,
Sure.
0.4 meters cubed. Thanks for all you do. I enjoy your podcast and very much the things surrounding the science. Never cut the banter. We couldn't if we wanted. All the best, Marcus.
So there you go. Now we know what a boatload is. Isn't that handy?
Yeah.
Pronounce
4 hectoliters.
It's just spelled differently.
hectoliters. I see.
Yeah, with a C. Yeah.
I see what it is. Yeah. Okay. Cool.
Rich, can you take the next one?
Charles writes, "Hello, Twivers. Wonderful 72 degrees Fahrenheit in Chapel Hill this fine Sunday morning. A couple of people that know me well have said I have an obscure sense of humor. That sense of humor is what caused me to refer to TWiV 1031 as TWiV 2007. It can all be explained with yet another Tom Lehrer song called New Math." I haven't looked through this song. Or I can just tell you that 1031 in base 10 is 2007 in base 8, and the song New Math does a math problem in base 8. Or even shorter answer, I am a Tom Lehrer nerd and wanted to know if some of you were fellow travelers. I'll bet some of your listeners have written in with the answer. Please give a count on the next TWiV. Thanks, Charles. Okay, so we puzzled over this 2007 thing last time and did not get it.
And Vincent was somewhat on the right track because
Yeah.
you said you tried it in base 2, right?
I did, yeah, but I didn't.
I count myself as at least somewhat a Tom Lehrer fan. My father had his first record, and I knew all of the songs. Okay, but I didn't follow through appropriately, so I don't know all of his other songs. I'm sure I've heard the New Math song, but not well enough to have applied it in this particular case. Vincent, did we get other Tom Lehrer New Math mail or 2007 mail?
No. This is the only one.
Well,
wow.
Charles and Marcus, you guys are spot on.
Thank you.
Okay,
Right.
You get it. All right.
That's right. I
mean, this whole show, I was thinking before we went on, this show is a nerd fest. I'll tell you. Okay. The phage paper and the Redondo virus paper. We are back to core essential TWiV.
So, Rich, did you actually memorize his Elements song?
No, I did not do that, and that was not on the first album. I don't think.
Oh, okay.
May not have been, but the old Dope Peddler. That's one of my favorites.
Poisoning Pigeons in the Park.
Poison. That was not on the first album either, I don't think. But the waltz song, I forget. At any rate, I knew quite a few of them.
Let me take the next one from Anonymous. I've been listening to your TWiV podcast since the beginning of the pandemic. I thoroughly enjoy all of the accurate scientific information that's presented by you and the team. I have an otherwise healthy 24-year-old daughter who was initially told she had a positive test for HSV2 taken from a lesion on her lip. Now she's being told she does not have HSV2. Her HMO has been reluctant to refer her to an infectious disease specialist to explain and clarify her diagnosis. This is very confusing and upsetting to her. Has all of the research around SARS-CoV-2 led to more research on HSV2? Is there a definitive test to test for HSV2? And is there more research being conducted on cures or treatments for HSV2 at this time? So I found an article about an mRNA vaccine to prevent genital herpes. HSV1 and 2 is part of the target. So you could take a look at that that's more recent research. And then you can get HSV2 testing. Try another lab. If, as Daniel would say, if one of them isn't pleasing you, go somewhere else.
Yeah. And to answer the question, has all the research around SARS-CoV-2 led to more research on HSV2? Probably not. Probably dollars have gone towards SARS-CoV-2, potentially away from HSV2 for the moment. But yeah. But there are definitely still people working on HSV2, and as Vincent reported, potential vaccine.
Jolene, can you take Darak?
Yep. Darek writes, Vincent has elaborated a conception of viruses where, if I've got this right, the virion is just one stage of viral life and the infected host cell is another stage. Did I get that right? Sounds good to me, but there's one implication and one comparison I was wondering if y'all could elaborate on. So I'll just stop there. The virion is one stage of the viral life and the infected cell is another stage. Definitely, we tend to separate those as different aspects of the viral replication cycle. Continuing, Vincent and other hosts make a point to correct statements where authors report estimating viral load with a molecular assay of the genome, such as qPCR. I get the feeling that a plaque assay would be preferred by this crowd, but doesn't a plaque assay just measure one part of the viral life cycle? Does viral load necessarily refer to infectious particles? Do infected cells count as viral load? Regardless, plainly reporting results in the context of the assay is critical for clear communication. So, doesn't a plaque assay just measure one part of the viral life cycle? No, actually, the plaque assay measures many parts, all the parts, the complete life cycle.
Yeah.
But in most cases, in order to
Even
generate a plaque, you have to infect a cell with a single active virus particle. So
Correct.
from an indirect point of view, it's a measure for the presence of an infectious virus particle.
Mm-hmm.
Of course, there's exceptions to all of this. There are some viruses where you need two particles, blah, blah, blah. Okay. And then you got to talk about, particle infectivity ratios and all of that. But I think plaque assay generally is looking at infectious virus particles.
Mm-hmm. And does viral load necessarily refer to infectious particles? Usually, yes.
But it could depend on the assay that's being reported.
Yeah, I think we tend to assume that viral load. I mean, that's kind of part of our definition, is that viral load, you're talking about active virus. The problem with PCR is you could be measuring bits of nucleic acid that are not part of a virus. Okay, so definitely PCR does not measure virus necessarily. Okay, it can measure other things as well. That's the problem, and that's why we equate viral load with plaque assays, where you're actually measuring virus.
I guess before the advent of genome measuring methods, we used infectivity, right? We used plaque assays or TCID50s.
Mm-hmm.
And so we were measuring infectious viruses. Then it got complicated by PCR, right?
And actually, I was just thinking about ways of quantitating virus nucleic acid, and you could have done that by Southern blot, or some kind of dot plot analysis.
Right. There was an equivalent. Yeah. One of the things I want to emphasize here, Darak, because this is, you get it, plainly reporting results in the context of the assay is critical for clear communication. Absolutely. That's the most important part of all of this.
Right. Yeah, I think that came up a lot. Oh, sorry, Kathy, go ahead.
No, you go ahead.
I think that came up a lot when you were talking about the measurement of SARS-CoV-2 genome copies in samples taken from humans. And there was questions about whether or not there were infected cells that were being measured, or lysed cells releasing nucleic acid that was being measured.
And that's a good example, because one of the things that everybody struggled with is that if you do PCR testing or nucleic acid testing over time during a SARS-CoV-2 infection, there's a huge long tail. And does that mean these people still have virus? Or is it just kind of leftover nucleic acid and the virus is cleared? So it becomes an important distinction.
Yeah. Yep.
And then I just want to point out that not every virus can be assayed by a plaque assay or a TCID50 if there's not a cell culture system in which cytopathic effect is caused by the virus, then you can't do that. So there are people who measure, for instance, norovirus loads from human enteroid samples, and you can't get CPE, cytopathic effect, in enteroids. So those assays are done by a PCR assay.
Yeah, that's the case for some phages as well, actually. Okay, continuing the letter. Second thing, I think I heard someone count the virus-infected cell, let's say while it's faithfully reproducing viable virions, as potentially being alive. Did I mishear that? I'm interested in this idea, in particular to wonder whether the viral infected cell, let's say it's actively producing viable virions, is a living cell or a living virus or both. So then, is a mitochondria infected cell a living organism? Are we chronically infected, perhaps even hijacked by mitochondria? Mitochondria are often, but not always, clonal and asexual. For exceptions, consider yeast. And Dirac gives a reference. Are mitochondria alive? Do we care? Let me just finish up here. I appreciate the philosophical bent amongst all the usual wonderful work y'all do. Keep it up, you know why. Dirac. And there's 2 PSs. PS, the double-headed the double-headed nanobody to target the FC-mediated functions reminds me of CAR T cells. Are a lot of future therapies just going hot-wiring the immune system to target whatever we want to clear? Neato. And PSS, looking for a long podcast? Try this 5-hour and 43-minute episode about aspartame. And that appears to be Stronger by Science, episode 116.
Wow. Sweet.
Oh. Virus-infected cells are alive until they're dead, right? I'm good with that. I'm good with that. But I wouldn't say that a cell is infected by a mitochondria.
Right.
That happened so long ago that it's now part of it, right? So,
yeah.
We had go back and listen to TWiV from episode 1, okay? Because there are many episodes in there where we struggle with this question of whether viruses are alive or whatever. We even took a survey at one point.
Mm-hmm. Did.
I didn't have a single question on that whatsoever. If a thing that you think is an organism does not have a metabolism, it's not alive. Period.
Yeah.
Viruses do not have metabolism.
Virus particles don't.
Sorry again?
Virus particles don't have metabolism, but virus-infected cells do. So.
Oh, yeah, right? Of course. But mitochondria have a metabolism.
So, it's interesting. There's a virologist, or his name is Bandea, Claudio Bandea. So he wrote this idea up years ago, this idea that there's a virus in this infected cell. They're both different phases of an organism, and the virus is not living, but the infected cell is. And I found it years ago, and I blogged about it. And he just wrote me last week, and he said, thanks to your blog post, I've gotten more attention than I ever had in my life. And he said, now people seem to accept this idea. And he said he wants me to write a Scientific American article with him that explores this. I thought it was funny.
That's a good idea.
But I really liked that idea. It was from him that there's this do phase thing, right? Because the virus particle has to eventually get into a cell, and then it takes it over, right? And it makes
sense
to me.
A cell is dead when it no longer metabolizes.
I don't know when it's actually dead, right?
Well, when it stops using glucose.
Well, if you turn off electron transport, it's dead, right? I think that's if you poison the chain, then the cell's dead.
That's right. If you disconnect it by unplugging it, it's dead. And that's what viruses do.
Because when someone dies,
there's a joke like that. We're not going to go there, but there is Mel Brooks tells a joke about that, but we're not.
A lot of things continue for a while, but anyway, let's finish.
Decay.
Kathy, I think you're next.
Okay. John writes, Dear esteemed Doctors TWiV, after 1031, when you mentioned complaints that you're engaging in politics, I had planned to write that it is not engaging in politics, when a politician first comes into your wheelhouse and declares that something you know to be true isn't, and you defend accepted science. I think you basically said the same thing in 1033. But then I ran across this semi-related quote from the famous 19th century agnostic Robert Ingersoll, whose birthday was yesterday, and which seems to illustrate how things used to be. Quote, "Now that science has attained its use and superstition is in its dotage, the trembling, palsied wreck says to the athlete, 'Let us be friends.' It reminds me of the bargain the cook wished to make with the horse. 'Let us agree not to step on each other's feet.'" Mr. Ingersoll would be dismayed at the rooster's tenacity now, some 140 years later, and with all of science's advancements in that interval to boot. Otherwise, we came off of a high 86 Fahrenheit, 30 degrees Celsius here in Greater Braddock today that wasn't that bad since the air kept moving. Now 72 Fahrenheit, 22 C this evening in the aftermath of a tornado watch and some scattered thunderstorms that only served to end the day's bricklaying efforts prematurely. And Vincent, I like the blue, green, yellow background in 1031. Cheers, John. So.
Okay, one more. Jim writes, love the show, been a listener since 2020, love learning about all kinds of viruses. With respect to the quote discussion on TWiV 1033 concerning the quote about how you can't use reason to get someone out of a position they didn't use reason to get into, there are versions of this quote going back at least to Jonathan Swift in 1721. Gives a link for that. That page also has many variations of this quote over the centuries, which, at least to my eyes, are slight rewordings of the same sentiment. Yeah, good sentiments have been around for a while, I guess.
I don't know if this is the same person that wrote to me directly, but somebody wrote to me directly with some more information about this quote. And
okay.
It goes way back. So.
All right, let's do some picks of the week. Dixon, what do you have for us?
He'd like to tell us, but he's muted. Dixon.
That's okay. One picture speaks 1,000 words. Last night I watched Cosmic Time Machine, which is a Netflix-based documentary, and it shows the evolution of how the James Webb Space Telescope came to be. And it has amazing footage, real footage, not fake. It's all from the telescope, and really shows how dedication and hard work sometimes produces a miracle. Science is the result. 10,000 people plus worked on that for about 20 years. And you can imagine how many times there were when, if someone made one small error, the mirror on the Hubble Space Telescope doesn't work, right? That was a small error, by the way. The calculation was less than the width of a human hair, and that was enough to cause that distortion. But nobody screwed up on this. Nobody. And that was the miracle. And this program documents that. I highly recommend it, even if you're not interested in astronomy. It's really amazing. And of course, this is the, 2-day-old picture from the Webb Space Telescope that you can see in front of you. It's called the Ring Nebula, and it's the aftermath. It's what's left over after a star burns out. And it's 10 times better in detail than the same picture from the Hubble. The process of star death, that's a supernova, is how all of the elements are created and then distributed throughout the cosmos. And this, if you blow this up, it's already blown up, of course, but if you blow it up so you can look at all the parts, you will be astounded at the detail that the telescope allows you. It's like the difference between a light microscope and an electron microscope. When those two, when there were only light microscopes, everybody was happy with the result. And the next thing you knew, along comes this new instrument, and you can look deeper and deeper and deeper into nature. Basically, you're looking into nature. And I'm absolutely taken by all of this new. I'm glad I lived long enough to see this happen.
Thank you, Dixon. I'm going to watch this. Looks great.
You will love it, Rich. You will absolutely love it.
Very nice. Beautiful. Kathy, what do you have for us?
I have something about Antarctican accents. So it turns out that, of course, Antarctic Antarctica doesn't have a permanent population. People go there transiently. But some scientists did a study in 2019 about whether there was a change in accents among 11 people who wintered over. They were recruited from the British Antarctic Survey. It included 8 people born and raised in England, 5 from the South, 3 from the North, and 1 person from the Northwest United States, another from Germany, and an Icelandic person. And they recorded their voices at the beginning of this study and made 4 more re-recordings about every 6 weeks or so. And at this time, of course, because they're wintering over, there's no influences from the outside, and these people are working closely together, socializing. And so researchers noticed a definite change in their accents. And one of the major changes was that people began pronouncing their words with longer vowels. There was evidence of linguistic innovation, and they were pronouncing OU sounds like in the words flow and disco from the front of their mouth as opposed to the back of their throat. So anyway, it's just a scientific study that they took advantage of people being in this situation in Antarctica. So there's maybe an Antarctic accent.
Maybe they picked up an accent from the penguins.
Yeah. That's very interesting.
Yeah.
That's cool. Very cool. Rich, what do you have for us?
So this is for you, Dixon. I was listening to NPR in my car the other day, and I was listening to the show is On Point, and they were interviewing a parasitologist, and I only heard bits of it, but the one bit I heard blew my mind, and it was about this parasite that's an isopod called Cymothoa exigua. Okay. And this is, what's an isopod? It's like a crustacean, right?
It is a crustacean.
Yeah, it's a little thingy, like
Yes, it
almost
is.
like a pill bug, only not really.
Yeah.
Okay.
Exactly.
This thing infects fish,
Right.
and the males, it starts off in the gills with small males that morph into females, okay, and wind up in the fish's mouth and latch onto the tongue and cut off the blood supply to the tongue. And so the tongue necroses and falls off, and this thing takes up residence and replaces the tongue and functions as a tongue. Okay, so the fish can't do without it from then on. All right, and so it's sitting there getting the first hit of any food that comes in, but doesn't compromise the fish necessarily. And I thought, man, this is great.
You can't make this stuff up.
You can't make this stuff up.
Make this stuff. You wouldn't want to either.
Make this stuff up. Yeah,
nobody would believe you. This is incredible, and I've never heard of that one.
You've never heard of that one? No, I actually taught Dixon something about a parasite. That's great.
So, Rich, everybody's happy in the end?
Apparently. I mean, they didn't take him to the opera, right? So I don't know.
Right.
They didn't interview him. Okay,
but
apparently
question.
the fish live, from what I can tell, a fairly normal life. But once their tongues are gone, they wouldn't be able to live without this thing. And there's some really nice pictures of the creature and the creature in the fish's mouth. And in fact, if you look at the link of the episode, there are even better pictures. Okay, there's a picture of a fish's mouth with this thing. 2 pictures of this thing sitting there. They're really amazing. This is why, in the greater scheme of things, this is personal. Okay, this is how I comfort myself when I start stressing about stuff like climate change. Okay.
All right.
Because DNA is going to keep replicating. All right.
Absolutely.
It's going to find a way. All right. Even if the humans are all gone, life will go on. All right. Because this kind of stuff is possible. Anything is possible.
Well, if the humans are gone, life will go on. It'll be good.
Yeah.
Without the humans.
Right. So, Kathy, the only thing that changed in the fish was that before the louse got there, they spoke beautiful, fluent English, and after that, their accent was really lousy.
Really good. Excellent.
Man.
Thank you, Rich.
Sure.
Jolene, what do you have for us?
I have a perspective article recently published in Cell. It is called Insights and Strategies for Improving Equity in Graduate School Admissions. And this is a project of an organization called Científico Latino. It's a graduate student mentorship initiative that they've been running since 2019 to support applicants from historically minoritized backgrounds to help them get into graduate school. And in this article, they detail a lot of the practices that they have used, a lot of the trends that they've seen for people who don't have good support communities to help them know what are the best practices as well as the common practices used for applying to graduate school. How can they improve their chances of getting in? Because that's just one step to being able to have good representation of all of us in this great science adventure that we're on. I'd especially like to point out figure 4, which is actually a table where they go through and detail recommendations to specific populations. And what I mean by that is in the first part, they say these are things that PIs, professors, and directors of undergraduate studies can do, regardless of what the rest of your institution chooses to do. I can go through I can put on my lab website, graduate school application resources. I don't have to make them up. I can link to resources that other people have developed and that are really well researched and referenced. You can put personal statement examples. There are different recommendations. The next section is for departmental efforts. They have things that you can put on your website, ways that you can help with financial equity. And then they have recommendations for institutional efforts, which are mainly targeted at financial equity. I think this is something that we can all play a part in helping make sure that the systems that we are participating in are as equitable as possible for everyone who has great contributions to give to our scientific enterprise.
So, Jolene, am I correct in assuming that this is all stuff that is possible, whether or not you can legitimately or legally in your institution have a DEI program?
Absolutely. There's nothing that says that you can't provide resources accessible for everyone to help them, guide them as they are applying to graduate school. And It's in fact encouraged to help students know what they should do when they're applying to your program. Mm-hmm.
So who's the target for this article? Is it PIs like yourself, admissions committees, or the actual students who would like some tips?
It's more about the people who can give the tips than the students themselves. Yeah. But it's nice to be able to read and see maybe yourself in a lot of the trends. It can be helpful to realize that you're not the only one that doesn't know what they're supposed to do or how you should ask for a letter of recommendation, things like that. Yeah, so
nice.
Lots of good tips in here for people to make things better. And maybe even if the people around you aren't making things better directly, you can reach out to those who can help you. And
it's open access. Good.
Yeah.
It's smart.
Yeah. And as Jolene pointed out, the figure 4 actually is a nice summary of sort of action items. And while I haven't read the whole article in its entirety yet, I did forward it on to several people. One already knew about it, but others were happy to learn about it. So I think spreading the word is good.
I'll also say for students who are looking for tips, if you're thinking about applying in this graduate admissions cycle this fall, the website of the organization that ran this graduate student mentoring initiative has lots of tips, and they can partner you with a mentor, and they accept anyone.
Oh, nice.
Great. Thank you. All right. My pick is a article in space.com. Why Chandrayaan-3 landed near the moon's south pole and why everyone else wants to get there too. So I heard people talking about this. India just landed a craft near the South Pole. Wednesday, I think Russia tried to do it and it crashed, right? And
Yep.
Russia's crashed.
And the U.S. is going to do it. China is going to do it. Why the South? Well, read the article. It's basically, we think there's water down there, and that could be useful to do a lot of things, make a base and so forth. So there are a lot of articles with similar bent, but that's the idea. I want you to think about why everyone's going to the South Pole of the moon.
There's a cool video in here that's a lander's eye view of the landing.
Right.
Yeah.
I have 2 listener picks. Douglas, this well-written article by Ben Hubert is likely to make interesting reading to you and all TWiVers. It's a very good article. I read it this morning. It is on climate change and active climate management. A really good, it's long, but it's putting out all the facts. What's going on with climate change? What can we do about it? Lots of good graphs. I liked it really a lot. So check it out. Thank you, Douglas. And Matt writes, Dear TWiV, apologies if this was already a pick. I haven't heard it mentioned. It sure seems up your alley. The topic and solution will come as no surprise to regular listeners of the podcast, but I found it a good reminder that we could reduce the likelihood of pandemics by leaving habitat for these animals and their ecosystems. And so this is in ProPublica. This scientist tracked bats for decades and solved a mystery about a deadly disease. This is Hendra virus, which got forced out of, the bats that carry it got forced out of their habitat by stuff that humans do. And so we could mitigate that to a certain extent. Although the article on climate change, it says, we could plant billions of trees and it won't make a dent because there are other issues going on. What's interesting in that climate change article? He said a real good thing to do would be to take a mountain and grind it up and throw it in the ocean. That would soak up a lot of CO2. But he said, should we be grinding up mountains? It's really a good article. You have to read it. It's really good. All right. Cool. Thank you, Matt. It's 68F and breezy this evening in Portland, Oregon. That's TWiV 1039. You can find the show notes at microbe.tv/TWiV. You can send your questions, comments, and picks to TWiV at microbe.tv. If you enjoy our work, please support us financially. Go to microbe.tv/contribute. Dixon Despommier, Racaniello.org, thelivingriver.org. Thank you, Dixon.
What an enjoyable, diverse, and exciting podcast this was. I truly enjoyed it.
Kathy Spindler, University of Michigan at Ann Arbor. Thank you, Kathy.
Thanks. This was a lot of fun.
Rich Conda is an emeritus professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Sure thing. Always a good time.
Jolene Ramsey is at Texas A&M University, ramseylab.vercel.app. Thank you, Jolene.
Gracias. I really enjoyed it.
And come back, and your lab is lovely. I just looked at your website. What a nice bunch of students and
they're a great bunch.
Postdocs. Lovely. Good for you, and good luck with that. I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.